Okay, so welcome to this next video in which we're discussing the acute inflammatory response and anti-inflammatory drugs. Okay, so we're still looking at the acute inflammatory response at the moment, and we're still looking specifically at type 1 activation of endothelial cells. Okay, so let's now look at the recruitment of neutrophils, which occurs by type 1 activated endothelial cells. Okay, right, so firstly, what's going to happen is that we've discussed how the phosphorylation of myosin light chain leads to the contraction of the endothelial cells, which opens up these gaps between neighbouring endothelial cells, which then allows the build-up of an inflammatory exudate, which brings in complement proteins, coagulation factors, and the components of the kallikrein-kinin system. But myosin light chain becoming phosphorylated also causes another thing to occur within the endothelial cells which have undergone type 1 activation. So let's have this as a endothelial cell here. So basically in normal endothelial cells which haven't undergone type 1 activation, what you have is vesicles within these endothelial cells, and this isn't a vesicle, this is the nucleus, but you have vesicles within this endothelial cell which will have proteins within them, okay? Proteins within the membrane of the vesicle. Okay, now I'll colour in this protein in purple. This protein is P-selectin. Okay, so in purple here, this is P-selectin, okay? And the name of these vesicles which store P-selectin within them is viable palade bodies. So they're so important that they have their own special fancy name, and they're called viable palade bodies, which I assume is named after the people who discovered them. Okay, so these viable palade bodies are a store of P-selectin in all endothelial cells in your body. So normal, non-activated endothelial cells do not have P-selectin on their apical membrane. Okay, instead it's all sequestered within these viable palade bodies. Now, when you get an increase in the level of phosphorylated myosin light chain, okay, so here is a myosin light chain protein with a phosphate group stuck on it. So this is myosin light chain in here, which I'll abbreviate as MLC, but I suppose I might as well write it out in full. So this is myosin light chain, which is now phosphorylated. What we know is that phosphorylation of myosin light chain leads somehow to the exocytosis of these viable palade bodies, and that this leads to the P-selectin being put on the surface of the endothelial cells. So endothelial cells which have undergone type 1 activation are going to display P-selectin on their surface. Now, they're also going to put another molecule on their surface which is going to be very important in the recruitment of neutrophils. And this is where we go back to the lysophosphatidyl-choline that we abandoned so long ago. Okay? So, you remember when we first started discussing type 1 activation of endothelial cells, we discussed that the rise in cytosolic calcium causes the activation of the cellular phospholipase A2, and that this causes the translocation of this cellular phospholipase A2 from the cytoplasm to the inner leaflet of the phospholipid bilayer, and the cellular phospholipase A2 then sets to work on the phosphatidyl-choline molecules in the phospholipid bilayer, breaking them down into lysophosphatidylcholine, this molecule here, and also arachidonic acid. Now, we focused completely on arachidonic acid, which was a star in this show. It was converted eventually into prostacyclin, which caused vasodilatation of the terminal arterioles. But now we're going to turn our attention back to this lysophosphatidylcholine, which is within the membrane of the the membrane of the endothelial cell. Okay, now what's going to happen to this lysophosphatidyl-choline? So let's just draw it out here again. So here is the choline, which we're just representing as this blue box, and I'll colour everything in again. So we had choline as this blue box down here. We had the phosphate group in purple here. Okay, we had glycerol in green here. Okay, and bound off the glycerol, bound to the first alcohol group of the glycerol, we had a free fatty acid. Well, not free anymore, a fatty acid though. Okay, so this was lysophosphatidyl-choline. Okay, so what's going to happen is we are going to stick something onto the vacated second alcohol group of this glycerol molecule within this lysophosphatidyl-choline molecule. So remember, we broke off the fatty acid that was in the second position, and now we've got a free alcohol group there that we can bind something to. Now we're going to stick on a very small carboxylic acid in replacement for that massive great carboxylic acid that we removed, which was arachidonic acid. We're going to stick on ethanoic acid, which the old name for which is acetic acid. Okay, now this is the acid which is in vinegar. Okay, so we're going to esterify acetic acid onto this lysophosphatidyl-choline, and this is done by an enzyme known as platelet activating factor acetylase, and that name doesn't seem to make any sense at all, because this enzyme is supposed to be adding an acetyl group onto lysophosphatidyl-choline, whereas platelet activating factor and then acetylase, and acetylase is something which removes an acetyl group from something. Okay, but this name makes sense if I tell you that it also catalyzes the reverse reaction. Okay, so the molecule you're going to get when you add an acetyl group onto lysophosphatidyl-choline, let's draw it here. You're going to get, here's the lysophosphatidyl-choline, and then you're going to add a tiny little carboxylic acid on there. Okay, so here is in orange these two carboxylic acids now. Okay, then we've got in green the glycerol molecule. In pink we've got the phosphate group, and in blue we've got the choline molecule. Okay, now this molecule is what's known as platelet activating factor, and for short this is abbreviated to PAF. So this stands for platelet activating factor. So this enzyme, platelet activating factor acetylase, catalyzes both directions of this reaction, and it's named obviously for the reverse direction where it's removing an acetyl group from platelet activating factor. However, it's also capable of catalyzing the forward reaction where it adds an acetic acid group onto lysophosphatidyl-choline and creates platelet activating factor. So these endothelial cells which have undergone type 1 activation, they're initially going to get lysophosphatidyl-choline in their membrane, but very quickly this platelet activating factor acetylase will convert this into platelet activating factor. So you're going to get platelet activating factor appearing within the phospholipid bilayer, and this is actually going to play an important role in neutrophil extravasation, which is something, the final thing that type 1 activated endothelial cells are going to do. Okay, so let's put this important molecule, well actually I won't put it on yet. Let's firstly discuss neutrophils. Okay, so the next star of our show, the neutrophil. Okay, so what was the point of the inflammatory response? It's always good to step back and look at what we were actually trying to achieve. So we were trying to bring in troops that were going to help deal with the pathogenic infection. So far, we have vasodilated the terminal arterioles to supply more blood to the affected area, and we've increased vascular permeability to let in a whole bunch of proteins. Complement proteins are going to help kill the pathogens, and also with the positive feedback reaction through the anaphylotoxins. Coagulation factors are going to help contain the pathogen, and the kinin cascade is just going to make the affected area painful so that we don't do anything that's going to make it worse, and also is going to lead to a positive feedback loop. What we really want, though, is to get some white blood cells, which are these specialised cells for destroying invading pathogens. We really want to get some of those in. Okay, and the first one that type 1 activated endothelial cells are actually capable of recruiting, that's going to come in, is what's known as a neutrophil. Now, neutrophils are pretty much the pawns of the immunological world, basically. They are a quite cheap cell, if you like. They are not a particularly powerful piece, but you have a huge number of them, so it doesn't really matter if you lose a few. Okay, so you're just going to chuck them at this infection. And they are a phagocyte, so they work by engulfing the pathogen, and hopefully, they will actually digest the pathogen down and destroy it. But that's a little bit hit and miss. Okay, so we need to get these neutrophils from the blood into the interstitial fluid so that they can then go and attack the pathogen. Okay, now, only type 1 activated endothelial cell, well, not just type 1, they're also type 2 activated, but only activated endothelial cells are going to do this, not normal endothelial cells. Normal endothelial cells will not interact with the leukocytes. And by the way, leukocyte is just a fancy word for white blood cell. Okay, so that just means white blood cell. Right, so a neutrophil is a type of a leukocyte. It's a type of white blood cell. So normal endothelial cells do not interact with the leukocytes at all. Okay, but now the viable Platelet bodies have fused with the apical membrane of this endothelial cell. Now we have P-selectin on the surface of our endothelial cell, and basically the neutrophils have a ligand for P-selectin. They have a protein which will bind to P-selectin. Okay, and this is given the rather sensible name P-selectin glycoprotein ligand 1. Okay, and because P-selectin glycoprotein ligand 1 is a bit of a mouthful, that's often abbreviated to PSGL1 for short. So P for P-selectin, well, P for P, and then S for selectin, G for glycoprotein, L for ligand, and then 1, PSGL1. Okay, so let's colour in PSGL1 in a blue here. Okay, so neutrophils have this molecule, P-selectin glycoprotein ligand 1, which will bind to P-selectin, which is now on the surface of these type 1 activated endothelial cells. Okay, now what is this going to cause? Well, it's going to cause the first phase of neutrophil extravasation. So let's go over the steps that occur in neutrophil extravasation. So step 1, in fact I'll put it up here, step 1 is a process known as rolling. So what's going to happen is the neutrophil is going to roll along the surface of the endothelial cells. And what happens as it rolls along the surface, it continually makes and breaks interactions between P-selectin and P-selectin glycoprotein ligand 1. So if you imagine this cell rolling over this endothelial cell, then when it rolls, it's going to break this interaction, but maybe it will make a new one. So maybe there is a P-selectin here, and then there is a P-selectin glycoprotein ligand 1 here. Okay, so this is PSGL, another PSGL1 here. So let me colour it in blue again. Okay, because you wouldn't be able to tell from their sizes that they were the same protein, but if I colour them in the same colour, then it will be obvious. Okay, and here is P-selectin in vivid purple here. So, what will happen is if you imagine this neutrophil rolling over this endothelial cell, it will break this interaction between P-selectin and P-selectin glycoprotein ligand 1. And of course, they're very, very weak interactions. And then it will forge a new one, and you'll just continue this process. It will roll along, just forging new interactions between new P-selectin glycoprotein ligand 1s and new P-selectins, okay? And this will gradually slow the neutrophil down. Okay, so let's now discuss the next phase. So, here is our next endothelial cell here. Okay, and the next phase that you're going to get is what's known as weak adhesion. Okay, now this involves another molecule which is permanently expressed on endothelial cells. So I want to stress that this is permanently expressed on endothelial cells, and it would not do anything without the new P-selectin and the platelet activating factor, which are essential for neutrophil extravasation. But this new molecule that we're about to see is permanently expressed on endothelial cells. So firstly, let's just say we will have an interaction between P-selectin and P-selectin glycoprotein ligand, but we're about to see a new interaction, basically. Okay, so it's come to a halt now, and we have a fixed interaction now between a P-selectin glycoprotein ligand 1, here in blue, and a P-selectin here in purple. Okay, so what's new? Well, the new bit is that you're also going to have an interaction between another molecule on the surface of the endothelial cell, and this is the one that's permanently on the surface of the endothelial cell, and this is what's known as, let me just move this up, this is what's known as ICAM1, and this stands for intercellular adhesion molecule 1. So the IC is for intercellular, the A is for adhesion, and the M is for molecule, and then 1. Okay, so the intercellular adhesion molecule 1. Okay, so let's colour in the intercellular adhesion molecule 1 in a certain colour. So we'll have it in red here. Okay, and it's a member of the cell adhesion molecules superfamily, which is a huge molecule of proteins which are capable of sticking to each other. So if one cell has a cell adhesion molecule on and another cell has another cell adhesion molecule on, they can join together, basically. And within the cell adhesion molecules, you split them into 4 main families. Okay, now one of the families is what's known as the Immunoglobulin superfamily cell adhesion molecules. And it is to this family that ICAM1 belongs. The other 3 families of cell adhesion molecules are the integrin cell adhesion molecules, the selectin cell adhesion molecules, of which P-selectin is a star, and the cadherin cell adhesion molecules. Okay, so ICAM1 is a cell adhesion molecule, and it's going to actually link to another cell adhesion molecule that is on the surface of the neutrophil here, and let's just make it obvious that it's a neutrophil by giving it this multi-lobed nucleus here. Okay, and the cell adhesion molecule that it's going to bind to is an integrin cell adhesion molecule known as LFA1, which is often referred to as LIFA1 for short. Okay, and this stands for, where should I write it? I'm not going to write it there because I want to keep some space there. Okay, well, I'll write it down here. So LIFA1 stands for the lymphocyte function associated antigen 1. Okay, so L is for lymphocyte, the F is for both function and associated, which are linked by this dash here, and then the final A is for antigen, and then it's 1, so lymphocyte function associated antigen 1. Okay, now let's colour that in turquoise here. Okay, so LIFA1 is permanently expressed on the surface of the neutrophils. ICAM1 is permanently expressed on the endothelial cells. Okay, they bind together. ICAM1 is an immunoglobulin superfamily cell adhesion molecule, whereas LIFA1 is a integrin superfamily cell adhesion molecule. Okay, and they initially bind very weakly, so this is what's initially known as weak adhesion, and it's because this integrin, LFA1, is not properly activated, so it doesn't form a very strong adhesion with the ICAM1 initially. Okay, now let me just discuss something about LIFA1. So basically it's an integrin cell adhesion molecule. So it consists of two subunits, an alpha subunit and a beta subunit. Now, when you're making an integrin, there are 18 different alpha subunits that you can use, and there are 8 different beta subunits you can use. Now, LIFA1 specifically consists of the alpha L alpha subunit and the beta 2 beta subunit. So it's what would be known as the alpha L beta 2 integrin. Okay, now, initially, this interaction between alpha L beta 2 integrin and ICAM1 is very, very weak. However, what's going to happen next is that you're going to activate that integrin, and the way that you activate the integrin is platelet activating factor. Okay, so the platelet activating factor, which is now in the cell membrane, and I'm drawing it as though it's a box here, but you must remember it's this which is within the cell membrane, so it's not something that's sticking out from the cell membrane, it's part of the cell membrane, and I'll just abbreviate it to PAF for short. Okay, so let's have platelet activating factor, I think in yellow, okay, or dirty grey as it's turned into, and there is a receptor for platelet activating factor on the surface of the neutrophil known as platelet activating factor receptor. So let's colour this in here in pink. Okay, so this is the platelet activating factor receptor. And basically, when platelet activating factor binds to the platelet activating factor receptor on the neutrophil, that will lead to the will lead to a downstream pathway in the neutrophil which will activate this integrin. It will activate this LIFA1 integrin, this alpha L beta 2 integrin. And suddenly now, this interaction between this LIFA1 integrin and this ICAM1 will suddenly become extremely strong. So it will go from being a very weak interaction to being a very tight adhesion. Okay, so you're going to get tight adhesion between the neutrophil and the endothelial cell. Okay, now the process that follows the final portion of this neutrophil extravasation is what's known as diapedesis or endothelial transmigration. Okay, so diapedesis is going to follow, and this is also called endothelial transmigration. Okay, so what's going to happen in endothelial transmigration is that the neutrophil is going to sliver through the gap between neighbouring endothelial cells. Okay, so let me show you this. So, if we have two endothelial cells here, basically, I have told you before that initially what happened is these endothelial cells were held together by tight junctions and adherens junctions that were between these neighbouring endothelial cells. Now, there was another junction that I didn't tell you anything about initially, okay, because it wasn't a particularly strong one, not compared to the tight junctions and the adherens junctions. Okay, so let me try and get my picture of the adherens junctions back again. So here, oops, not there. Here we have our tight junctions and our adherens junctions between two neighbouring endothelial cells. Now, even below this, below the adherens junction, there is another junction, okay? So let me put these two membranes here. Okay, so this is the membrane of endothelial cell 1, and this is the membrane of endothelial cell 2. Now, below the adherens junction, you then have another junction, which is between two cell adhesion molecules, one on endothelial cell 1 and the other on endothelial cell 2. And these cell adhesion molecules that are on the two cells are utterly identical, and it's a cell adhesion molecule known as PCAM1. Okay, now this stands for, where should I write its name? I'll try and squeeze it in here. So it stands for the platelet, that's the P, and then the E is endothelium, or rather endothelial, and then cell adhesion molecule is CAM. And then it's the platelet endothelial cell adhesion molecule 1. And it's again a member of this immunoglobulin superfamily cell adhesion molecules, and that fitted perfectly. Okay, and what's going to happen is both endothelial cells will have a pCAM1 molecule on their surface. So here's the pCAM1 molecule from endothelial cell 1, and then here is the pCAM1 molecule from endothelial cell 2. They will bind together, and that helps to hold the two endothelial cells together. Now, what's very important about pCAM1 is that what the next process of neutrophil extravasation is that the neutrophil is going to slither like a snake through the gap between the two endothelial cells, which involves the neutrophil deforming quite a lot to fit through that very small gap. Okay, now as it slithers through this gap, it has pCAM1 molecules on its surface which will be interacting with the pCAM1 molecules on both endothelial cells. So when you got endothelial contraction, like the tight junctions and the adherens junctions were pulled apart, the pCAM1 junctions were pulled apart, and now the neutrophil also has pCAM1 on its surface, so it will form pCAM1 interactions with both sides of these of this gap. Basically, it'll form interactions with this endothelial cell and this endothelial cell, and it's these pCAM1 interactions that help to hold, helps to sort of negotiate the neutrophil through this gap. So let me just draw this. So here's the membrane of endothelial cell 1. Okay, here is our sliver of neutrophil coming through, and here's the membrane of endothelial cell 2. So you're going to form these interactions between pCAM1 molecules with both sides, basically. So you'll form pCAM1 interactions with the membrane of endothelial cell 1 and also with the membrane of endothelial cell 2. Okay, so neutrophils have pCAM1 proteins on their surface which help in this slithering through the gap, which is known as diapedesis. Now, what's then going to happen is these neutrophils will go into the interstitial space, and they will then go and phagocytose the pathogens and hopefully help to clear the infection. Okay, now in type 1 activated endothelial cells, the only type of white blood cell that you are capable of recruiting from the blood and moving into the interstitial fluid to any great extent is the neutrophil. Okay, we're going to have to wait for type 2 activation, to be able to recruit more types, basically. And we'll continue this discussion where we'll look at type 2 activation next in the next video.